So three scientists rediscovered individually on the Mendel's experiment, and then after that two scientists came. Can you give me the names? Walter Sutton and Boveri, right? Sutton and Boveri. So previous lecture by mistake chapter number 8 લખાઈ ગયો. 4 સેમ ની અંદર chapter 8 છે આવ્યો. એટલે ખાલી મગજમાં આવે નથી લખવું. જેને સમજ હોય જેનેટિક્સ સમજો. હા very good. Now Coran's give two laws of Mendelism. According to the 1 MCQ, I think you are confused behind this. First law of Mendel is law of which law? Dominance. Yes, the first law of Mendel is law of dominance, and second law is known as law of segregation. And law of segregation is also known as law of purity of gamete. Later on I will explain this. Okay. So Coran's gave the two laws. Laws of inheritance or heredity or Mendelism. Understand this completely? Perfect. First law is known as law of segregation. Known as law of purity of gamete. Anyone, can you explain this law of segregation? Anyone? Plant consists so many characters. In heterozygous condition, the two gametes for two different characteristics present in the same plant. I am just talking about capital T, small t, heterozygous tall plant. But when you think about that capital T, small t in that heterozygous condition, when the gametes are formed, You know that one capital T and one small t. So plant consists both the genes. Now I am using the word genes. Plant consists both the genes for tallness as well as dwarfness. But formed gamete consists only and only one, that is perfect character. Either capital T for specifically tall plant or small t for dwarf plant. Dwarfness means. Similarly, if you are talking about the C, capital A, capital B, small b, according to the monohybrid, okay, but here I selected two characters. Think about capital A is whatever, small a is whatever. You have that 7 contrasting traits of the Mendel, right? So think about that capital A is this and small a is this and capital B is this and small b is this. So if you are talking about the monohybrid one, fine, if you consider only one character, 4 gametes are formed, capital A, small a, capital B, small b. If you had learned the dihybrid experiment properly, so in dihybrid experiment, at the time of the gamete formation, I had told to you that the gametes formed from the P generation, 4 in number, type is only 1. Number of gametes are 4, but type is only 1. Here, see, if you consider only one character, number of gametes are 4 and types are also 4 because 4 are totally different from each other. Okay, again, capital A, capital B, small b, you take it. Now you consider dihybrid one from this. So, if I will tell you that capital A can be taken as a first, small a is second, capital B is first and small b is second. So now here you know that 4 gametes are formed, capital A, capital B, capital A, small a, capital B, small a, small b. So here we say like that, first, second, first and second. This trick I had already given at the time of the dihybrid experiment explanation. So here similarly, you see that the number of gametes are 4, types are also 4. And here same way when you consider the 2 characters, number of gametes are also 4 and types are also 4. You think about capital R, capital Y, capital Y. Form the gametes. Yes, 4 gametes are formed. That all the gametes consist capital R, capital Y. So we say that number of gametes are 4, but type is only 1. Okay, now you tell me. Capital R, capital Y, small y. How many gametes are formed? And how many types of gametes are formed? Can you got the question? Okay, I can write this. How many gametes? Second, how many types of gametes? Answer is totally different. Tell me. Number of gametes are 4, but type is 2. Why? because the heterozygosity is there only in Y, right? Okay, so type 2. Same way. Same question. How many gametes are formed and how many types of gametes are formed? See, this is tri-hybrid. This is tri-hybrid. 3 characters. Tell me, think about it. No. Number of gametes are 8 or 6. Someone says 6. Someone says 8. Answer is 8. And how many gametes are there? 4. Because heterozygosity is there in the 2 characters. Clear? So same way, here you think about that. I will give you the clue. Okay, later on I will tell you that. But according to this much information, I will give you the clue like that. Number of gametes are formed. It is in the manner of 2 raised to n. N means number of contrasting traits. Number of contrasting traits. So if you take only one contrasting trait, one character, so 2 gametes are formed. And if heterozygosity condition is there, then 2 types and 2 number both are same, like this. Clear? And in the homozygous condition, in that gametes are same. Clear? So, law of segregation is also known as law of purity of gamete. Here, organism consists the mixture of characters. But at the time of the gamete formation, only and only one, and that is specific character that is obtained in the gamete or present in the gamete. Right? That's why this law of segregation is known as law of purity of gamete. Then, yesterday I told to you that law of dominance. Law of dominance explain that what is dominance and what is recessive. The definition you have to give it here. And along with that, according to the law of dominance, in monohybrid experiment, in F2 generation, how 3:1 ratio came. That explained by law of dominance. Right? Second law is known as law of independent assortment. See, if in board exam, the question is asked like this, explain dihybrid cross method. and 4-mark question. So in textbook, very brief information is given. Only chart is given there, nothing can be explained there. Only chart is given there in dihybrid experiment. So in the dihybrid experiment, you have to write about the law of independent assortment also. So this law of independent assortment is the law of dihybrid experiment. And monohybrid experiment, in the 2 laws are there, law of dominance and law of segregation. But here Correns gave the 2 laws of inheritance. Pea plant is annual plant with short lifespan 2 to 3 months. And due to this small lifespan, less lifespan, along with that it is easily cultivated in the garden. And as you know that in the 2nd chapter, you will learn with the emasculation and bagging method. So actually here the garden pea plant, Pisum setarum plant, it consists bisexual flower. Bisexual flower or monoecious flower, or you say that hermaphrodite flower, all the words are same. So to prevent the self-pollination, we have to do emasculation. And emasculation, can you define it? Removal of anther with the help of 2 forceps without damaging any other wall of the flower. In the bud stage, proper word, that is bud stage. In the floral bud stage, because anther જ્યારે મેચ્યોર થઈ જાય ત્યારે એને રિમૂવ કરવાનો મતલબ ન રહે. ઓકે, અહીંયા તમારે ખાસ શું ધ્યાન રાખવાનું છે કે anther release the pollen grain and stigma become receptive. Before this phenomenon, we have to remove the anther at all. So in the floral bud condition itself, with the help of the 2 forceps, without damaging to the other walls of the flower, carefully remove all the anthers from the flower, and this method is known as emasculation. Another twisted thing, emasculation is nothing but the bisexual flower, it is converted into female flower. And as you know that in androecium, the main important part is androecium. In androecium, main reproductive part is anther. Anther is important. So in androecium, the periphery anther is important, and in gynoecium, the proximal ovary is important. Remaining parts are sterile. Yes, filament. There is no role of the filament in the fertilization procedure. No role of connecting. No role of the stigma. No role of the style. That's why these all are accessory reproductive organs. Helpful in reproduction, yes, helpful in reproduction, but no role in reproduction. Please understand the words properly. Fine. So, pea plant is annual plant with a short lifespan, 2 to 3 months. Now, you have that table in the textbook, 7 characters, contrasting traits. You remember that table properly, but at last I will give you the conclusion. So in table you edit it. In table sideways space is there. So you little bit elongate the table on the sideways by making the heading on number of chromosome. Otherwise, wherever space is there, you edit it. Now I can tell you the type of the character which is situated on the which number of the chromosome. Clear? કયું કેરેક્ટર કયા ક્રોમોસોમ ઉપર છે એ તમને મારે કહેવાનું છે. So wherever you want to write it. Length of the plant, stem height, it is situated on the 4th chromosome. Flower position, 4th. Yes. Shape of the pod also on the 4th. Again, the length of the plant stem height, the 2 contrasting traits, alleles are present on the 4th chromosome. Flower position on the 4th chromosome. Shape of the pod also on the 4th chromosome. Pod color on 5th chromosome. Seed shape, 7th chromosome. 2 are remaining, right? Which? Seed color and flower color. Both are present on the 1st chromosome. So might be in Gujcat. Might be in Gujcat. And in NEET, the question will ask like that. Which type of the question came? What do you estimate? Mendel has selected the 7 contrasting traits or 14 characters of the Pisum sativum plant are situated on how many chromosomes? 4. These all 7 contrasting traits or 14 characters are present on the different 4 types of the chromosome, and the specifically numbers are 1, 4, 5 and 7th number. Clear? Another conclusion. two characters, two contrasting traits are situated on the first chromosome. Two contrasting traits are situated on the first chromosome, and three different contrasting traits are present on the fourth chromosome. Fifth consists 1 and 7 consists 1. So out of these 4, you first remember the 5th and 7th because it has only one contrasting trait. And you remember the first chromosome which has 2. Only 3 you remember it. And how the MCQ will come? Fine. કયા ટાઈપનો MCQ આવશે? See, straightforward તો 1, 4, 5 and 7 number of chromosome के 4 types of the chromosome, 14 types of the characters are situated on the 4 types of the different chromosome. Same way. Information તો તમને આપી દીધી. Specifically particular trait છે, contrasting trait, which is situated in the which number of the chromosome, that will also ask. Sometime mark odd one out. કઈ રીતે mark odd one out આવશે? You have the answer. See, mark odd one out. Length of plant, flower position, seed shape and shape of the pod. Seed shape. Why? Because now how you make this odd one? Yes, according to the location on the number of the which chromosome. Fine. So 3 characters are present on the 1 chromosome. This seed shape, it is present on the 7th number of chromosome. So that is odd one out. Clear? ઘણી વખત આમાં તો તમારે 2 ફ્લાવર કેરેક્ટર આપ્યા છે, 2 સીડ કેરેક્ટર છે, અધરવાઇઝ એમાં બી માર્ક ઓડ 1 આવી શકે છે. Clear? So this is about the monohybrid experiment. Now you write next point. obtained wrinkled seed. Mendel obtained wrinkled seed due to absence of starch branching enzyme SBE. Actually, you will learn this in biomolecule. Starch is polysaccharide, and polysaccharides are divided into two types. Unbranched polysaccharides known as amylose, and branched polysaccharide is known as amylopectin. So here in seed of the pea plant, due to absence of the SBE, free sugar molecules are present inside the seed instead of the starch. Clear? So please remember this thing. Wrinkled seeds are due to SBE absence. Okay, now you see monohybrid experiment. Phenotypic ratio, it is also known as basic ratio or Mendelian ratio. Phenotypic ratio is also known as basic ratio or known as Mendelian ratio. And see, in the F2 generation, the phenotypic ratio is 3:1 and genotypical ratio is 1:2:1. So in these characters, the capital T and small t are obtained. It is known as impure varieties. So sometimes the question will be asked like that. In monohybrid experiment, what is the proportion of the impure varieties? 50%. Right? So 1:2:1 is there. Now see, the conclusion of the monohybrid cross. First conclusion is postulate of the paired characters or factors. So first conclusion is that characters are controlled by factors and factors are always in pairs. Second conclusion, postulate of the dominance. When two heterozygous factors or traits are present together, but here main important thing, no